The overall goal of the following experiment is to image the neuronal activity of the mouse with Manganese-Enhanced MRI. It is done by unilaterally stimulating the vibrissae and mapping the neuronal response in the contralateral barrel field cortex. The first step is to assemble and calibrate the ultrasound system, consisting of an unfocused ultrasound transducer driven by the signal generator and amplifier. For the experiment, after an intraperitoneal injection of manganese, the blood-brain barrier is opened with ultrasound and microbubbles, allowing the manganese to enter the brain parenchyma. Next, the mouse is subject to a neuronal stimulation paradigm, which results in higher accumulation of manganese in the more active regions of the brain. Finally, MRI images are obtained that show regional differences in T1-weighted MR image intensity based on different levels of manganese accumulation within the brain.
In neuroscience, the mouse is the dominant model system for studying the genetic and molecular bases for brain development and disease. This technique provides a non-invasive tool for mapping neural activity in these mouse models.
Not only can ultrasonic disruption of the blood-brain barrier be used for delivering manganese to the brain parenchyma for the purposes of functional neuroimaging, but it can also be used to deliver a wide variety of other compounds into the brain parenchyma, including gadolinium-based contrast agents or chemotherapeutics.
Generally, individuals new to this method will have difficulty calibrating the ultrasound system and handling the microbubbles.
Demonstrating the procedure will be Yi Chi, one of the technicians in our laboratory.
The ultrasound system consists of a single-element ultrasound transducer